or columns, which are known as columns, functional columns. There are about 1 million of these columns in the human brain. Each of these columns is about 0.5 millimeter in diameter. And one can debate forever about what is a column. But the bottom line is a column is the minimum set of neurons that create a society for neurons, because neurons are not islands. They need a group of neurons around them. And the minimum set of group of neurons around them turns out to be approximately the size of a column. In the rat, it's about 10,000 neurons. In humans, it's about 100,000 neurons. In evolution, these columns were added until we ran out of space and they had to keep folding in on the brain. And that is one of the reasons why the brain is so highly convoluted. We just kept adding these chips. So the secret to the neocortex is to understand these little columns. And the goal of my lab in the past 10, 12 years has been to go inside these little columns and try and reverse engineer how are they built with exquisite detail, and as much detail as we had technology that would allow us to do. So just to give you an idea what that looks like, this is a technique that we developed at that time in Bert Sakmann's lab. It's called multineuron patch clamp. You put a slice of the brain, you put it under a microscope, you use infrared optics to visualize the brain while it's alive, this piece of the brain. You use a glass pipette to approach the neuron, clearing the way by positive pressure, and then you patch on, you suck onto the tip of the membrane, and you then blow a little hole in the membrane, and then you have electrical continuity. You can record the electrical behavior. You can inject a dye into it so you can see the physiology, the morphology, and you can record from many of these cells and start sending little messages between them. This is an extremely powerful technique because for the first time it gave us the tools that we can systematically start reverse engineering how the microcircuit is built. So over the years we've developed this to the most extreme art that is possible right now with the technology. And this is a 12-patch setup. We can't get more manipulators in there. We need to have very fine submicron precision of control of these manipulators so that we can choose these cells. But this has allowed us to rapidly increase in the rate at which we get the data to work out how the circuit is built. This is just what it looks like if you now inject a dye into these and you use a confocal laser scanning microscope, and you can see what is the full structure of these cells. So the first thing that you hit upon is you rediscover Cajal's inspiration that he must have had when he looked under the microscope. And that is that, and he called them the butterflies of the cerebral cortex, is that you have a whole spectrum of different types of cells. You have, it's the same, you go into the Amazon forest, you have a whole spectrum of trees. And here you do the same. This is a daunting variety of trees, and when you go to the computational people and you show them this is what it is, they of course are completely flabbergasted. How are we going to understand the brain with all these different types of neurons? Well, it turns out that they do cluster and group into certain classes, and basically there are 9 types of what we heard in the previous lecture as inhibitory cells, and there are also about 9 or 10 types of excitatory cells located in different layers. So there is an order, and it is not that complex. It's just that each of these types are different. And of the 100 billion or 30 or 100 billion neurons in the brain, there's no neuron that is the same. It's like people. There's no person that is identical, even for identical twins. No neuron is the same, but they do fall within classes. So we can now model these cells, any one of these cells. And this is an example. What we have to do is very often we need to repair first. Before we model them, we have to repair them because in the slice, we may shave off some of their branches. And this is just showing, for example, an algorithm that we use to repair the dendrites, which are in red, where neurons receive, and the axons. Once you've studied 100 pine trees or 1,000 pine trees, You don't need to get every 100 billion one of them in the brain. You can clone them. So we can essentially now clone as many neurons as we need of each of the types with the full morphological diversity that we see in the neocortical column. So we have the neurons. The next thing is we need to be able to capture the electrical behavior. We inject, when you inject positive current into these cells, this is a positive step current. The cell, of course, depolarizes. It's called depolarizing because all cells, including plant cells, sit at about -70, -80 millivolts resting potential, and then suddenly they spike. And these are the action potentials that is thought to be the currency of the brain, thought to be the basis of perceptions and consciousness. Neurons produce action potentials. But if you look at the neocortex or any other part of the brain, you discover another big challenge, and that is that they all have different behaviors. Not only do they look differently, but they also have different behaviors. And we need to now understand these different behaviors. Some of them have stuttering behavior, some of them wait before they start firing, and some of them have bursting behavior, and so on. We've characterized most of these different behaviors, like a psychologist, I suppose, characterizes all the different patients. And what we've seen is that each of the anatomical types can also have multiple electrical types. So you may have a certain anatomical, a pine tree, but that pine tree may have different behaviors. And if you put that together, you start to see that in the neocortex, in one layer, you have many different types of neurons. These are the names of all the different types of neurons that you find in one layer. It's about 50 types. So you're looking at about 300, over 300 types of neurons in the neocortical microcircuit. We can capture them, each one of them again. They may fall within a class, but they all are slightly different. So not only they're structurally different, but they're also electrically different. So now our challenge was we need to be able to model not only the classes, but all the different variations within a class. So what we had to do is to start going further below the surface and find out exactly why they're different. Now we know that the neurons are different because they have different behaviors, because they use ion channels. They have ion channels for sodium, calcium, chloride, potassium. And there's in the neocortex, there's about 200 ion channels that they can select from. And if I select 20 of these, I'm going to have a certain behavior. And if I select 20 of those ion channels, I'm going to have a different behavior. So we have to work out now the 200 ion channels. We have to be able to model each one of them. And then we have to know which one corresponds to the different behavior so we can build each of these neurons. So over the past 10 years, one of the techniques that we've used and developed was to go into these cells and extract the cytoplasm, the juice from inside. And it's like a reverse copy process. We take the messenger RNA to reverse copy a complementary DNA of the genes that are switched on in that single cell. So we can use primers to find out which genes are switched on in that cell. These are names of ion channels that are used, and we can see that these genes are switched on to produce those ion channels in that one cell. Now it gets more complicated because these genes produce subunits, and these subunits can combine in a combinatorial manner to give you over 200 ion channels. So we've had to work out how they combine, what types of ion channel combinations are used in the neocortex, and we've mostly done that and been able to, we start now to build a large database of these different types of ion channels, and we know now which ones of these we need to use to model each of these different types of behaviors. But it's not that simple. It gets a little bit more complicated, and that is